Hello, welcome to another video. Today we will be doing the diffusion through a membrane lab, pre-lab for part 2. So we already did the diffusion through a membrane part 1 lab where we looked at the model cell and the movement of starch and glucose through a model cell. In part 2, in the lab that we're going to do this week, we're going to be looking at osmosis of water in response to a change in concentration of solutes outside the cell. And the solute that we're going to look at is salt. So let's go ahead and get started. All right. So we're going to go ahead and look at now the pre-lab. And today's aim is how can we prepare for the diffusion through a membrane state mandated lab. So this is again one of the state mandated labs. So this is very important for passing living environment. All right. So, and this lab is going to be about osmosis, which is the diffusion of water. All right. So, when preparing a wet mount of onion cells, a student put a drop of Lugol's iodine solution on the slide. Lugol's iodine solution, you might remember we use Lugol's not only as a starch detector, but also to stain the slides in the first microscope lab we did. So Lugol's iodine solution is a stain. And why do we apply a stain to the cells? It's to make the cell structures more visible. Question number 2, a diagram to the right shows how a cover slip can be lowered onto some single-celled organisms during the preparation of a wet mount. And so this is a technique that you guys would be perfecting during this lab. Unfortunately, we cannot do it in the actual lab, so you won't be perfecting the wet mount, but this is a wet mount. And why is this the preferred procedure? It's to prevent the possibility of trapping air bubbles. All right, so, oh, and there's an image of making a wet mount slide. So in this lab, you will prepare a wet mount slide. In this lab, you will prepare a wet mount slide. You will also need to label all the organelles you see. For this lab, you need to make sure to take a sample of onion that contains the red pigment. So we use red onions to do this lab, and this is fundamental to understanding the concepts of the lab, because if you don't use a piece of red onion where the red pigment is visible, what will happen is you're not able to observe the change in the size of the cytoplasm that occurs in response to osmosis. So, all right, part B, review. And also, yeah, so we don't add stain in this lab. We just use water, which is why you need to take a part of the onion that contains the pigment. So a movement of molecules from an area of high concentration until equilibrium is reached without the use of energy is called diffusion. And we should hopefully all know that in our sleep by this point, right? Diffusion is from high to low. We know that's a diffusion. All right, so now we're going to describe or draw a typical red onion cell mounted in water. So we're going to write a couple of things about the red onion cell just first. Okay, so the red onion cell, when it's in water, the cell is turgid, meaning, remember we talked about plant cells. Plant cells are made primarily of water. They have that cell wall that makes the plant cell structure right? More rectangular and more sturdy than an animal cell, for example, because we have skeletons for our structure. And when the cell is mounted in water, it is plumped up full of water. So what does that look like? Let's go ahead and draw it. So we're going to have the outside of the plant cell, and because it's a plant cell, the one that we're going to look at here is not going to be green. It looks almost like purple colored, so I'm just going to do this in black and white. I'm not going to do it green like we would normally do a plant cell. But the separation between these structures is important, so that's why I went back and did that. All right, so here is the red onion cell mounted in water, and now we're going to label some structures. So we're going to label the cytoplasm, and in this case, in this lab, we'll see the cytoplasm, and it's shaded like a pinkish, purplish color, and that's how you can tell where the cell, what the cell membrane is doing, because inside of the cell membrane is obviously the cytoplasm, right? So then in this area, let's go back here. We just said it, right? So what is this? That borders the cytoplasm. That's the cell membrane. So it's across the cell membrane that diffusion and osmosis occur. And then the last structure that makes the plant cell unique and is the outer border here is the cell wall. So then this space between the cell membrane and the cell wall is still basically the cell wall. All right, so the movement of water from an area of high water potential to low water potential, so where the concentration of water is higher to lower, is called osmosis, which is the diffusion of water. Osmosis. All right, part C, onion cell and salt water now. So we already talked about what happens in response to water. Now what happens in response to salt water. Salt water is a solution containing both salt and water. So this means that the concentration of water is lower in salt water than it would be in regular water. Where there is a high concentration of salt, there is a low concentration of water. Water will then move towards the area that has a high concentration of salt in order to balance out the system, right? So if there's more salt, that means there's less water. So now we're going to describe and or draw a typical red onion cell mounted in salt solution. All right, so now we're going to draw the red onion cell when it's mounted in salt water. So remember, when it's mounted in salt water, the water from the cell is going to be moving towards the salt, right? The water is going towards the salt because there's a lower concentration of water where there's more salt. So when it's mounted in salt solution, the cell membrane and cytoplasm shrink. as water leaves the cell. The cell membrane retracts further from the cell wall. So let's draw what that looks like. So we have here again the same sort of rectangular plant cell structure. But then within, instead of sort of copying the outside of the cell wall, once the salt water has been added, the cytoplasm can take on a jagged shape, right? And that's because water is leaving the cell. So water, as it leaves the cell, as it leaves the cytoplasm, right? Because the cytoplasm is this jelly-like structure, it's dehydrating it. And you can see this really well in the slide because when you use, and this is why you use the onion that has the red, when you use the red onion, the cytoplasm is this nice red color and you can see it shrunken down. And we'll look at images of this obviously and watch a video when we do the lab. But so now let's label this cell. We've got cell wall, which is still the outside of the cell, but now the cell membrane way retract from the cell wall, right? Normally they're close to touching each other. Now it's retracted, excuse me. And why did it retract? It's because so much water left the cell. And it gives us a good view of that jelly-like structure within the cell membrane, the cytoplasm, which is where all that water was that left the cell. Water. Via osmosis. And why did the water leave the cell? It's because we put all the salt on the outside of the cell. So inside the cell, there's a higher concentration of water. Outside the cell, there's a lower concentration of water. So the water inside the cell leaves via osmosis because it's going from high to low. And of course, the salt, if the salt was not too big to diffuse, it might go into the cell, right? Because the concentration of salt is higher outside the cell. So that can be confusing. But we know that salt is too big. Salt molecules are big molecules, right? So the salt isn't going into the cell, but rather the salt outside the cell is causing the water to leave the cell via osmosis. And that's the process that we're demonstrating in lab part 2. So when salt water was added to the wet mount, when salt water was added to the wet mount, the cytoplasm appeared to shrink in size. This is because water left the cell since the salt concentration is higher outside the cell, right? The salt is higher outside the cell, but, and it's important to remember this, concentration of water is higher inside the cell. So that's why the water moves, because the water oh, I'm trying to draw that water droplet, but not succeeding. The water is still going from low to high, because osmosis, or excuse me, from high to low. The water is still going from high concentration inside of the cell to low concentration outside of the cell. Because even though we added salt water, right, that has a low concentration of water because there's so much salt in it. So the water is still moving from high to low via osmosis. All right. So now last cell drawing. And what you can do for yours, if you're not able to draw, is you can Google red onion cell mounted in distilled solution, red onion cell mounted in salt solution, etc. And you should be able to find images and you can paste them in. So let's get to drawing the last one. So in this case, it's in distilled water. So again, we're going to be looking at a turgid cell. So what happens in lab when you do this is you start off with a cell in the regular wet mount, and then you add salt. And so when you add the distilled water, the cell at first is all like, blah, 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 right? And then over time, right, because diffusion, osmosis, it takes time. Over time, as water flows back into the cell, right, via osmosis, because you've added this distilled water solution. So what the distilled water solution does is it puts a higher concentration of water outside the cell, meaning water flows into the cell. So over time, that little shrunken cytoplasm puffs back up to all her beautiful glory. And she's shining and living her best life. And for a final time, let's go ahead and label that cell. Ladies and gentlemen, we've got coming in, very important to transportation in and out of the cell. We've got cell membrane, beautiful, exciting, critical to the removal of waste. We've got, oh, that arrow could go a little bit deeper in. We've got the jelly-like substance that supports all of the organelles within the cell. We've got our cytoplasm, which is plump and filled with water right now. And then on the outside, giving structure to the plant cell, making her unique, we've got the cell wall. And that is our cell. And again, it is turgid, water diffused back into the cell. Because there was a higher concentration of water, high concentration of water, and then inside the cell, because the salt had taken the water out, there was a lower concentration of water. So then what happened is the water flowed in, and that's osmosis. And when distilled water was added to the wet mount, the cytoplasm appeared to grow in size. This is because water entered the cell. Since water concentration is higher outside of the cell. All right, ladies and gentlemen, so today we talked about how we can conduct part 2 of the diffusion through a membrane state-mandated lab that is all about osmosis. I hope you guys enjoyed that. Please don't forget to go ahead and complete that exit ticket. And tomorrow on Zoom, we will be conducting the part 2 of the diffusion through a membrane lab. See you guys all there. Have a super fantastic day. Like and subscribe to my channel. All right, bye guys.